Surface functionalization of calcium phosphate nanoparticles via click chemistry, covalent attachment of proteins and ultra-small gold nanoparticles. Welcome to Journal Club. The latest academic research, explained in plain English. Today we're looking at this paper from MDPI's Journal of Chemistry. It's authored by Koska, et al., from the University of Duisburg-Essen in Germany, and is published open access under a CC BY license. Let's start by translating the title into more accessible language. When you read this title, you can understand it to mean in very simple terms that this paper explains a way to attach proteins and tiny gold particles to calcium phosphate nanoparticles using a chemical method called click chemistry. Let's expand on that a little. The research paper investigates a method for modifying tiny particles made up of calcium and phosphorus atoms. This modification involves using a chemical process called click chemistry to attach proteins and very small particles made of gold to the surface of the calcium phosphate particles. This could be useful in medical applications, as the modified particles could potentially be used to deliver drugs or other therapeutic agents. Now we understand the title, which is a good start, but before we can break down the full paper, we'll need to catch up on some terminology, jargon, background information and context. Specifically, there are some key concepts you'll need to be familiar with. Let's define them now. Calcium phosphate nanoparticles. Very small particles made up of both calcium and phosphate, commonly used in biomedical applications due to their biocompatibility and ability to deliver drugs or genetic material to cells. Click chemistry. A type of chemical reaction used to attach small molecules to a larger molecule, such as a nanoparticle, for various purposes including drug delivery and imaging. Polyethyleneamine, PI, a type of polymer commonly used in biomedical applications due to its ability to bind DNA and proteins. Carboxymethylcellulose, CMC, a water-soluble polymer commonly used in drug delivery due to its biocompatibility and mucoadhesive properties. Silica shell, a layer of silica added to nanoparticles to improve stability and facilitate further modification. Azide/alkyne groups, chemical functional groups commonly used in click chemistry for covalent attachment of molecules to nanoparticles. Hemoglobin, a protein found in red blood cells that is responsible for transporting oxygen throughout the body. Bovine serum albumin, BSA, a protein commonly used as a model protein in research studies due to its high abundance and structural stability. Ultra-small gold nanoparticles, very small particles made up of gold, commonly used in biomedical applications due to their ability to penetrate cells and interact with biological molecules. Dynamic light scattering, a technique used to measure the size and distribution of particles in a solution based on the way they scatter light. Now that we understand the title and key concepts, we're ready to dive in and break down this paper. In this presentation we're going to attempt to answer 5 questions. 1. What were the researchers trying to figure out? Or more specifically, what was their hypothesis? 2. How did they conduct their experiment? That is, how did they test their hypothesis? 3. What were the results of the experiment? 4. What conclusions did the authors draw from those results? And lastly, 5. Why does this research matter? Why should you care? 1. What were the researchers trying to figure out? The researchers hypothesized that they could covalently attach proteins and ultra-small gold nanoparticles to calcium phosphate nanoparticles using click chemistry, and that these functionalized particles would be well dispersed in water, amorphous, and easily taken up by cells without causing cytotoxic effects. 2. How did they conduct their experiment? The researchers used various reagents and instruments to synthesize and characterize calcium phosphate nanoparticles with different surface functionalizations. They used centrifugation and purification techniques to isolate the nanoparticles and determine their size and properties through DLS, SEM, XRD, UV-Vis spectroscopy, and EDX. They also attached azide and alkyne groups to the particle surface and conjugated proteins and gold nanoparticles to the functionalized surface using click chemistry techniques. The researchers characterized the resulting functionalized nanoparticles using various spectroscopic techniques. 3. What were the results? The researchers quantified the number of cargo molecules attached to the functionalized calcium phosphate nanoparticles using UV-Vis spectra, and Lambert-Beer's law, as well as calibration curves. They were able to determine the loading number by calculating the molar ratio of cargo molecules to nanoparticles. For ultra-small gold nanoparticles, the ratio of calcium to gold was similar, and fewer gold nanoparticles were conjugated to negatively charged calcium phosphate nanoparticles due to the presence of glutathione on the surface of the gold. The number of AF488 molecules clicked to the particles did not depend on the particle charge. 4. What conclusions did the authors draw from their results? The researchers concluded that conjugating proteins to the surface of calcium phosphate nanoparticles via click chemistry leads to a stable attachment, as shown with model proteins hemoglobin and bovine serum albumin. This process can be extended to attach other proteins and can also be used to coat large calcium phosphate nanoparticles with small gold nanoparticles, creating stable superparticles. The synthesis must be fine-tuned for every protein or potential cargo molecule being attached. It is possible to attach more than one species to the calcium phosphate surface, but careful purification, labeling, and analysis are needed for full characterization. Overall, this research provides pathways for the synthesis of multifunctional calcium phosphate particles for various therapeutic purposes. 5. Why does this research matter? The research paper demonstrates a versatile method for attaching cargo molecules such as proteins and ultra-small gold nanoparticles to the surface of calcium phosphate nanoparticles. This opens up pathways for developing multifunctional calcium phosphate particles that can transport cargo molecules into cells without toxicity. The stable covalent attachment of cargo molecules via click chemistry is a significant achievement that can be extended to other proteins, antibodies, immunostimulatory proteins, and nanoparticles. The ability to attach more than one species to the surface of calcium phosphate nanoparticles opens up possibilities for developing advanced drug delivery systems with multiple components. We hope you've enjoyed this Journal Club presentation of surface functionalization of calcium phosphate nanoparticles via click chemistry, covalent attachment of proteins and ultra-small gold nanoparticles. If you'd like to read the full paper yourself, which we encourage you to do, you can find it at DOI 10.3390/chemistry 5020072.